So now we are going to see the steps. First one is binding and fusion. So beginning, the HIV virus enters into the host cells. It firstly enter into the CD4 cell. So where the CD4 cell occurs, in the sense it is the T helper cells or helper T cells, which is mainly found in our white blood cells. So HIV begins to enter into your CD4 cells by binding or attaching itself to a specific point, that is they used to attach with the one receptor. That receptor may be of CCR5 co-receptor or CXCR4 co-receptor. So this allows the virus to merge with the CD4 cells. That process is called fusion. So after fusion, HIV releasing its genetic material, which is RNA, and the enzymes into the CD4 cell. So here the HIV's genetic material RNA, which contains the instructions to be followed, and that RNA will reprogram the CD4 cell so that it produces more number of viruses in our human body, and that HIV's RNA must be changed into DNA with the help of reverse transcriptase enzyme, and the newly formed HIV DNA enters the nucleus in the CD4 cells, which is the process of integration. And another HIV virus called integrase combines or integrates HIV's DNA with CD4 cells DNA. So in transcription, only the virus is integrated into the CD4 cells, and these proteins are the building block of new HIV virus. And assembly, when coming to assembly, the HIV enzymes, it cuts the long chain of HIV proteins into smaller pieces, and the smaller pieces came together with copies of HIV RNA, and the new virus is put together in the form of chains. And budding, this is the new assembled buds out of original CD4 cells. So this virus can now target and infect the other CD4 cells. And these are the diagrammatic representation of the above mentioned 6 steps. Coming to pathogenesis, it is excreted in blood and body fluids like semen, vaginal secretions, breast milk, cerebrospinal fluid, amniotic fluid, and synovial fluid. And it infect helper T cells, monocyte, microglial cells, oligodendrocytes. So under pathogenesis, you have the following headings, that is mode of transmission, primary infection, dissemination of virion to lymphoid organs, clinical latency, elevated HIV level, and clinical disease and death. So coming to the mode of transmission, it is transmitted through sexual contact, parental route, and congenital route. And second one is primary infection, that is acute infection, where it is the starting stage of disease. Only it is detectable in 8 to 12 weeks after the HIV virus entered into our host cell. And dissemination is virus from the blood then disseminated throughout the body to lymphoid organs. And clinical latency, it is an immune response which we gave for HIV, develops 1 week to 3 months after infection, which leads to significant drop or decrease in viral load. And this period of clinical latency is as long as 10 years. And the elevated HIV level, it leads to the symptoms of opportunistic infections like tuberculosis, candidiasis. And the very good symptoms of HIV is fever, sore throat, weight loss, night sweat, rashes, nausea, meningitis, encephalitis. And clinical disease and death, the duration will be in the primary infection to clinical diseases 10 to 12 years, while death occurs within the 2 years after the AIDS is in untreated case. So these are all the graph represented CD4 count wherein the HIV status is done by weeks and in years. Thank you.